All right, everyone, we're moving on to our final part of Unit 4 area of Study 2, which is basically us finishing VCE biology. So in the last lesson, or the last couple of lessons, we looked at different things that apply DNA knowledge, so gene cloning, genetic screening, and DNA profiling, as well as creating genetically modified organisms and how they are used mostly in agriculture. And then this last bit that we're going to look at here covers a few dot points, but it's really focusing on disease and how, we use biological knowledge with disease. So when we're talking about modern disease management, we're looking at how we can, using technology and understanding, identify and prevent infectious disease outbreaks in human populations. Now, obviously, you're all very familiar with disease outbreak in the past year and a half. But just a bit of a recap from Unit 3, Area of Study 2. So infectious or transmissible diseases are caused by pathogens, and these can include bacteria, viruses, fungi, protozoa, and parasites. Okay, so we're kind of talking about pathogens in general at the moment. Other things that are just worthwhile keeping in mind, pathogens often exist for very, very long periods of time before causing disease. And then, what kind of often results in them starting to cause disease is that they might mutate, meaning that they become more virulent or more infectious, or they may jump from an animal host to a human host. And that's what we see quite often with viruses, and COVID is an example of that. It was living for probably a really long time in animals, and then it jumped to a human host. Also just important to remember that there are multiple different modes of transmission of pathogens. So some are transmitted through water, some are through direct contact, some are through droplet or airborne, some are through indirect contact. So again, we covered those things in Unit 3, Outcome 2. All right, so just looking at actually classifying disease outbreaks now. So this term epidemiology is the study of diseases, particularly the study of the spread of diseases. So their incidence, how often they are occurring, the distribution, so where they have spread to, the cause of them, so what particular pathogens and how they are spreading around, as well as how to control these diseases. So epidemiologists have had a very, very busy year, in 2020 and 2021. Some specific terms that you need to know the definitions of. So if a disease is considered endemic, that is basically the term we use for a disease which occurs at a regular or stable amount. So for example, having just normal levels of chickenpox. It's not something that has been eradicated, but there are just certain levels of it that we consider to be endemic. And that generally means that it's controlled enough that it can be treated and managed and not kind of overwhelm systems. And then outbreaks are when you get an unexpected infection of large numbers of people. And again, we are very familiar with the term outbreak at the moment. Within outbreaks, there's two specific terms, and you do need to know the difference. So an epidemic is when you get a sudden increase of a disease in a specific population in a specific location. Okay, so that usually would be like in a city or in a country, would be considered an epidemic. A pandemic is when a disease spreads to different countries and or continents. And the word pan means all, okay? So just think of it that way. A pandemic is something that spreads all around the world. Again, obviously COVID is a great example of that. It began as an epidemic, okay? So when it was just in Wuhan, that would have been considered an epidemic, and then it became a pandemic. So just really important you know the difference between those terms. You don't need to know any examples of specific, epidemic or pandemic outbreaks, but, it is worth noting that these things are not new. These have happened throughout human history at fairly regular intervals, and they have, been very, very devastating, a lot of them in terms of the numbers of people that have died. But we now probably for, the first time, I guess, really in modern history, have very strong systems to manage disease outbreaks. And while I know you all probably feel that, this has been an enormous disruption on your life, and it absolutely has, we have these systems in place to protect from disease. Okay, so we're going to start by just looking at a few public health measures. So these are probably things that seem a bit less scientific, and you should be familiar with all of these, but the study design does say you need to know, strategies to prevent the spread of diseases. So some of the most common factors that would cause disease spread are things like poor hygiene and sanitation. Okay, strategies to reduce that are obviously things like hygiene practices, so washing your hands, having gloves, hand sanitizer, and personal protective equipment, so things like masks. Having very densely populated areas obviously makes it much easier for pathogens to spread. So that's why things like social distancing and isolation have been brought in those densely populated areas to try and prevent that human-to-human transmission. Increased global travel. Obviously, that's only something that's really happened in the last 50 years or so, and that poses a huge risk to things spreading. So that's where things like quarantine or health screening come into play to try and prevent these things from spreading. Certain diseases have very long incubation periods, which is that time between infection and showing symptoms. So if a disease has a long incubation period, it means a person can spread it quite easily without knowing that they have it. Okay, so for diseases with those long incubation periods, having lots of testing, and we've seen that obviously with COVID, get tested as early as possible, or if you think you might have been exposed to something, contact tracing. So that's being able to, if someone has been diagnosed, being able to identify people they were in contact with, because again, they might have the disease but not realize because they don't have any symptoms. And then, of course, isolating people that are at risk who probably were identified through contact tracing. Another one, and this is a bit different, or I guess it's nothing to do with COVID, but the overuse of antibiotics is a really big disease threat in terms of the fact that it has created really high numbers of antibiotic-resistant bacteria. So they are bacteria where antibiotics are no longer effective or won't work on them. And we'll talk about antibiotics in a little bit. But a strategy to try and decrease the rise of these antibiotic-resistant bacteria are things like having really strict protocols on antibiotic use. So, you can only get antibiotics from a doctor prescription. You can't just buy them for anything. And, aiming to reduce the use of antibiotics in livestock, because huge amounts of antibiotics are used in the farming industry, and that can contribute to this issue. Okay, so you just need to have an idea of different strategies that are used to reduce disease transmissions. All right, now we're going to look more at the more scientific level, and starting by methods that are used to identify pathogens. It's extremely important that we identify a pathogen that is causing disease, especially if this is a new disease, for a few reasons. Identifying the pathogen helps scientists to understand the behavior of that pathogen. So how is it spreading? How is it causing symptoms? What's that incubation period? How does it impact body tissues? All of those kinds of things. Having an idea of the pathogen and its behavior also allows scientists to work on methods to prevent it, and the main one being vaccination. Vaccinations are different for every different pathogen, so you need to have a really good understanding of the genetic components of a pathogen in order to create a vaccine for it. And then, of course, identifying the pathogen allows us to treat it. There's two main methods that are used to identify pathogens, and both of these would be used in modern science. The first is physical methods, which is, just visualizing the pathogen with microscope. It's worth noting that this is mainly used for bacteria, as viruses are very, very, very small and much harder to visualize well with microscopes. And then there are the molecular methods. So this is basically, looking at the genetic structure of a pathogen. It's DNA or RNA. And the two main methods for that are using PCR, polymerase chain reaction, to amplify particular segments of the pathogen's genetic material, or whole genome sequencing, which is getting the entire genome, all of the DNA or RNA bases, and analysing those. All right, we're going to focus the rest of this lesson on different ways of treating pathogenic diseases, so diseases that are caused by pathogens. And we're going to look at three different things. Antibiotics. Antibiotics are used for bacterial infections only. Okay, they will not work on a viral infection. Some antibiotics are specific, meaning a specific antibiotic is used for a specific type of bacterium. Other ones are what we call broad spectrum. So there are some antibiotics that might be effective against 20 different types of bacteria. Okay, so that's why it's really important that you only ever get that you get antibiotics with a prescription, because you could be taking antibiotics that aren't even going to be effective against the type of bacterial infection that you have. Antivirals, as the name suggests, treat viral infections. These are mostly specific, so mostly specific to certain viruses, but there are some that have a more broad spectrum action. And then finally, we're going to look at this thing called rational drug design. And this is the process of creating medications that are designed to act on certain pathogens. So these are always specific to certain pathogens and their molecules. So we'll start with antibiotics. Again, they only work on bacteria. Okay, make sure you know that. Questions have come up before about why would antibiotics not be effective against the influenza virus? Because they only work on bacteria. There's a few different mechanisms of action that antibiotics will have. This term, antibiotic sensitive bacteria, basically just means normal bacteria that don't have resistance to antibiotics. Okay, so when we're talking about the action of antibiotics, we're assuming that we're talking about antibiotic sensitive bacteria that will be destroyed by these antibiotics. There's a few different mechanisms of action. So one is that the antibiotics will inhibit the cell wall synthesis. So when bacteria are dividing, remember you have a bacteria to go through binary fission, it'll create two more, they will create two each. So in the process of those dividing, antibiotics can interfere with the cell wall forming, and that means that bacteria will not be able to function properly. They can inhibit the replication of DNA. So again, if a bacteria cannot reproduce its DNA, it can't divide, it can't go through binary fission. So it won't be able to spread in that area. They can inhibit protein synthesis. Okay, so that will impact the function of the bacteria if it can't produce proteins. And then similarly, they can inhibit the activity of enzymes. So again, that will prevent the functioning of that bacteria. Okay, so there are four different ways that antibiotics can work on bacteria. As I've mentioned before, some bacteria have resistance to certain antibiotics. So they are called antibiotic resistant bacteria, and they are able to kind of bypass these functions of antibiotics. Up here, this is just an example from a past SAT question. This is showing a plate. It's hard to see, but 4 different antibiotics were used. So all of this blue represents bacteria, a bacterial colony, and then there's 4 different antibiotics that were used, and it was left for a few days, and this is what was observed. So you can see around A hasn't really made any impact. There's still all that blue around there. Around C, a tiny bit of white. Around B, no impact. But around D, we've got this big section of white, which means that the bacteria there have been destroyed. Okay, so this antibiotic is effective on this particular type of bacteria. All right, antivirals. And again, if you have forgotten the action of viruses, it's worth looking back at Unit 3 outcome 2 and looking at how viruses operate. But the important thing is viruses need host cells. Okay, a virus can't do anything unless it can get into a host cell and use that host cell's machinery. So antivirals basically work in a few different ways to prevent viruses using host cells. So the first way they can act is to prevent, so these here are the viruses, and this is the host cell here. So this could be your cell. First thing that an antiviral could do is that it stops the virus getting into the host cell in the first place. If viruses can't get into cells, they can't do anything. Other antivirals will, the virus might still enter the cell, but it won't allow it to release its DNA, that viral DNA. Again, if it can't release its viral DNA, it can't use your cell to replicate. Another action is that they could prevent the actual replication of the DNA. Okay, so stopping the virus making more copies of its DNA, or they can work by preventing the formation of a new capsid. So remember, the capsid is that kind of shell around a virus. Without the capsid, it's just floating genetic material. It's not going to be able to travel. So preventing that capsid would mean that the virus is non-functional. Okay, so 4 different actions of antivirals. All right, the last thing is looking at rational drug design. So this is the process of developing medications that are specific to a biological target. It involves designing small molecules that act as ligands. So remember, ligands are molecules that bind to bigger molecules. We looked at these with cell signaling. So this is kind of making artificial molecules that can act as ligands. And what those ligands will do, or what these molecules will do, is bind to other molecules that could be part of a pathogen, or it could be something like an enzyme, and inhibit their function. Okay, so very similar in action to enzyme inhibitors that we looked at in Unit 3. What's really, really important is that these ligands that are kind of produced have to be complementary in shape and charge of the protein or the enzyme that they are binding to. So let's say, for example, we've got this enzyme here, and we're going to look at a specific example in a second, but we've got this enzyme, and this is the substrate that would normally fit into that enzyme. It's complementary. When that substrate fits into that enzyme, certain reactions will occur. If this is part of a pathogen's activity, what we want to do is stop this substrate binding with the enzyme. Okay, so what rational drug design would do is create this molecule that this end bit is actually the same as that substrate. Okay, it's complementary to the active side of the enzyme, and now this molecule can bind with the enzyme instead and block the active site so that the substrate can't bind with it. Okay, so that's why it needs to be complementary in shape and also in charge. What that means is if this enzyme here was positively charged, the molecule would be negatively charged. A specific example of this is a drug called Relenza, and this is specified in the study design. This was an Australian designed drug, but Relenza is an example of a molecular drug, so it was made using rational drug design that is used to treat the influenza virus. So when someone has the influenza virus, there's an enzyme called neuraminidase. It's a long name. Neuraminidase is the enzyme that stimulates the release of the influenza virus from a host cell. Okay, so remembering that, a virus will use a host cell and then make lots and lots and lots of copies of itself. Okay, neuraminidase allows those copies to get released from the host cell after they have been made. You can see that here. Neuraminidase comes along and basically releases these viral particles. Obviously, that just means that that virus is spreading further in the body. So Relenza, this drug, binds to the active site of this neuraminidase, the neuraminidase enzyme. So you can see here, this here is Relenza. That has bound to the neuraminidase enzyme, and now that enzyme can't bind with those viral particles and allow the cell to release them. Okay, so by inhibiting the binding of neuraminidase with these influenza viruses, the virus can't leave the cell and it can't spread. Okay, keep in mind at the same time as this, an immune response would be occurring that is targeting all of these cells to try and destroy these cells. So this is basically stopping, the spread of the virus further within the body while the body undergoes its normal immune response to get rid of that pathogen. Okay, so it is important you know how Relenza works. So how it would have been designed by creating that complementary molecule, complementary in shape and charge molecule to the neuraminidase enzyme, and that by blocking or by binding with the active site of that enzyme, it prevents neuraminidase from allowing the influenza virus to leave the host cell. Okay, so that is it for today, and that is it for Unit 4 Biology. So we've looked at strategies that deal with the emergence of new diseases, identifying pathogens, we've looked at antibiotics and antiviral drugs, and also rational drug design, specifically looking at Relenza. So have a go at the quiz questions, and that is it. Thanks.